good day. Now I'll be discussing to you about the inhibiting the growth of pathogens in vivo using or through antimicrobial agents, which is the chapter 9 in the book Microbiology and Parasitology, Borton's edition. So we have the so-called chemotherapy. Every one of us are familiar with this word. So chemotherapy is the use of any chemical or drugs which is used to treat diseases. So under chemotherapy underlies the antimicrobial agent, which will be the main topic or the main word that I'll be discussing to you throughout this video. So when we say antimicrobial agent, it is the chemicals or drugs that is being used to treat infectious diseases. So antimicrobial agent works mainly on these two functions. So first, it could either inhibit the growth of pathogens, or second, it could kill bacteria. So it's just two. It inhibits or stop the growth of certain organisms, or it could kill. So under the antimicrobial agent, we have the antibacterial agent, the antifungal agent, the antiprotozoal agent, the antiviral agent. So don't be confused with this these four, because when we say antibacterial agent, it is for bacterial diseases. Antifungal agent is for fungal disease, of course, and antiprotozoal agent is for the protozoal disease, and for the antiviral is for the viral diseases. Now, we have another word, which is the antibiotic. So take note that all antibiotics are antimicrobial agent, but not all antimicrobial agent are antibiotics. So when we say antibiotics, it is the substances which are being produced by certain microorganisms. So microorganism itself can produce or are able to produce these antibiotics or substances which has the possibility to kill other microorganisms. Now, why is this happening? Why do we have to kill why do certain microorganisms have the capacity to kill or inhibit the growth of others? It's for survival. As we all know, in survival, take for example, there is a shortage of food, so those fit or certain microorganisms must survive through killing or destroying the other so that they will benefit from it. So that's the answer about why this all happened, why there is a need for a certain microorganisms to destroy other. So that's it for antibiotics. So the example of antibiotics are penicillin, cephalosporins, bacitracin, which could be found in the book or if you search it on the Google. But take note that antibiotics are produced by first molds and second bacteria. So penicillin and cephalosporins are usually produced by molds. Meanwhile, certain bacteria produce antibiotics. So antibiotics in this advanced era, so some antibiotics are being manufactured or synthesized in the pharmaceutical laboratories. So we have the so-called semi-synthetic antibiotics. So what does it mean? So semi-synthetic antibiotics are those chemically modified antibiotics. So when we say chemically modified, some parts of it or some parts are being changed, being improved or enhanced or being developed. Take for example the ampicillin and nafcillin. So ampicillin and nafcillin are semi-synthetic penicillin. So what is the use of modified antibiotics? So if antibiotics are chemically modified, they reduce, or they have the lesser chance or lesser side effects. And another is that once it is chemically modified, it has a wider range or a wider scope or degree of killing pathogens or the disease causing, or when we say pathogen, it is the cause of certain diseases. So in semi-synthetic antibiotics, it is being developed to have a wider extent of killing pathogens. So that's it. So what are the characteristics of an ideal antimicrobial agent? So first, it must kill or inhibit the growth of pathogens, as just what I have been explaining earlier. Second, it should not cause damage to the host. Why? Because that's why we have antimicrobial agents, in order to treat diseases or those pathogens. So it should not cause damage to the host. Like take for example us. We animals and humans can be a host for these microbes. And when we say ideal antimicrobial agent, it should be good. Another thing, it should not cause allergic reaction to the host. We should take for example, antifungal antifungal agent. Since fungi is a eukaryotic cell and we humans are also eukaryotic cells, so there are certain circumstances wherein we are being when we are having some reaction to the drugs or certain substances that is being taken by us, because we are both eukaryotic cells. So that's why it is ideal for an antimicrobial agent to not cause any harm to the host. Another is that it should be stable when stored in solid or liquid form. So that's self-explanatory. Stable in order to remain in specific tissues in the body long enough to be effective. So it should last longer. It should survive in any condition, may it be in solid or liquid form, may it be in hot or cold. And lastly, it should kill pathogens before the pathogen can mutate or reproduce themselves or become resistant to those antimicrobial agents. So it's very ideal that before the pathogen could even start to reproduce, it is being destroyed or killed by the antimicrobial agents. So this is very ideal. Um, so there are 5 common mechanism actions of antimicrobial agents. So how does antimicrobial agent work or function? So it inhibits cell wall synthesis. So when we say cell wall synthesis, it stops or it hinders or it blocks the formation of cell wall. So take note that cell wall are for prokaryotic cell. We do not contain cell wall, but instead we contain cell membrane. So that's why the next common mechanism action of AMA or antimicrobial agent is it damages the cell membranes. Take for example the fungi, the protozoa. They are eukaryotic cells. So if bacteria have cell wall, then fungi and protozoa or the likes have cell membrane instead of cell wall. So the antimicrobial agents destroy the cell wall or cell membrane synthesis. Next, inhibition of nucleic acid synthesis. So when we say nucleic acid synthesis, this is where the DNA and RNA resides or are located. So as we all know, organisms cannot reproduce without this DNA or the genes, right? So that's it. Antimicrobial agent also inhibits or stop the mutation or reproduction of fungal certain pathogens because of this. And lastly, it inhibits the enzyme activity. Since enzymes when we have activities of enzymes could also result into a what do you call this one, synthesis of certain molecule, I mean of a certain cell formation. It is needed in the chemical reaction of the cell. So that's the 5th common mechanism action in antimicrobial agent. So we have the so-called bacteriostatic drugs and the bactericidal agent. So when we say bacteriostatic drug, it is the main function for this is it inhibits the growth of pathogens. Meanwhile, when we say bactericidal agent, it literally kills. It does not just inhibit, but it kills the bacteria. So another terminologies that we should be familiar with is the narrow spectrum antibiotics and broad spectrum antibiotics. So from the word itself, narrow and broad, narrow spectrum antibiotics are those antibiotics that could kill only one type of bacteria. It could either be gram positive or gram negative. Meanwhile, for the broad spectrum antibiotics, it has the capacity, or it is capable of killing both gram positive and gram negative bacteria. So we must take note that in applying bacteriostatic agents, we should be aware of the defense mechanisms of the host. Why? Because we should learn how to differentiate if the defense mechanism of a susceptible host do function properly, or their defense mechanism, or take for example those leukopenic or immunosuppressed patient, or infected organism. We should be aware of that. Why? Because if it is most it is much better to apply or put bacteriostatic agents to those, for example, us humans. We cannot just put bacteriostatic agents to those immunosuppressed individual, because when we say immunosuppressed, their immune system are being hindered or being blocked, or their immune system are weak. Take for example those persons that are having chemotherapies and such. And another thing is those leukopenic patients. Take for example, when we say leukopenic, it has a low count of, or low number of white blood cells. So white blood cells are the cells capable, or the cells that is that functions mainly on fighting or protecting our body against viruses, against pathogens. So if a certain patient lacks or has a low WBC, so it is not recommended. We should be more careful with in terms of applying bacteriostatic agents, antibiotics and such. So we have this so-called multi-drug therapy. When we say multi-drug therapy, we use 2 or more drugs simultaneously, because there are certain diseases that are being resistant to only to one drug. So we should apply more drugs in order to kill the pathogen in that disease. And we have the so-called synergism and antagonism. So synergism, it is the use of two drugs, two antimicrobial agents, because if we in synergism, from the words of synergist, so the two drugs could when they are combined, they work more effectively than rather than being taken one by one, or each drug is taken alone. Meanwhile, antagonism, it means two drugs work against each other, so they are not compatible with each other. Take for example two physicians recommended certain medications. So this, for example, this is the medication 1 and this is the medication 2. This is the medication given by the physician 1 and this is the medication given by the physician 2. So these two drugs might not, if combined together, they are not compatible. So they're not compatible, they're against each other. So in antagonism, it is better it is more effective if one drug is being used alone rather than being combined to the other one. That's antagonism. So, lastly, we have the so-called drug resistance or the superbugs. So, when we say the superbugs, it is those organisms resistant to antimicrobial agent. And we also have the MDRO or the multi-drug resistant organism. So when we say superbugs, organism resistant to only one antimicrobial agent. One. They fight, they are resistant, they are not being affected by a one certain antimicrobial agents. Meanwhile, for the multi-drug, from the word itself, multi-drug resistant organisms, it pertains to those organisms that are resistant to different kinds of antimicrobial agent. So how do bacteria become resistant to drugs? Well, of course, we must take note that there are certain bacteria that lack specific target site for the drug. Take for example the mycoplasmas. They have no cell walls. So how could an antimicrobial agent penetrate to the cell if they don't have cell wall in a prokaryotic cell? And remember that the common mechanism action of an antimicrobial agent is to inhibit the formation of cell wall or the cell wall synthesis. So that's how bacteria become resistant to drugs. So I hope you learned something from my video.